things I like so much about this picture is that Sherry is really doing mentoring there, and she's mentoring her daughter. And her daughter was interested in absolutely everything that was going on, absolutely everything. And it was wonderful to see how you could engage somebody so young in something that was so complicated. Not the complication of the launch, which is complicated enough, but on the subject that Sherry is going to talk to you about today, which is astrobiology. And this gives me a thrill because 12 years ago, I asked Sherry if she would be the editor of a journal called Astrobiology, and she had never edited a journal before, but she became the editor-in-chief of the journal. She still holds that position. And it's all about, are we alone, life in the universe, evolution, and the questions have implications for all of the work that we are doing here, and all of the work that you are doing has implications for the field of astrobiology. Would you say, Sherry? Okay, so Sherry is right now the Chief Science Officer of the Environmental Molecular Sciences Laboratory. And the rest of her bio, of course, is here. And she had been at Portland, where she was at Portland State University in Oregon as a full professor. And I was always envious of the field trips that she took, and I still am. But I think you're going to find this presentation absolutely of tremendous interest and one you haven't heard before.
Thanks, Marianne, for having me, for Carla making arrangements and everything. I'm not going to show a lot of statistics. I really don't know the statistics about women involved in the field of astrobiology. I kind of am probably not at that stage in my life where I think I'm avoiding it for my own reasons. I want to keep charging through. And I don't really want to know how bad it is, but I want to keep inspiring women and making sure that they have opportunities, the same crazy opportunities that I was able to have. I have to thank Marianne because she's been a wonderful mentor these last 12 years with the journal Astrobiology. I had no idea what I was getting into. And what I learned is that it's been one of the most pleasant opportunities that I've ever had in my professional career. Being able to find out what scientists are doing in the field up front and helping make that happen and helping them be successful has been just very exciting for me. I'm going to talk a little bit today about astrobiology. NASA is one of the federal agencies that has, I think, really started to understand how to connect with the public, how to take knowledge about the science that's going on, the missions that are going on, and help the public become aware of what all that is about. I also want to talk a little bit about the search strategies for what we're doing at Mars. We currently have a mission on Mars, and then I'll talk about some examples that come from some of the research that I'm interested in. So what is astrobiology? As Marianne said, it is a field that's focused on life's origins, evolution, and distribution of life in the universe. It's very interdisciplinary. It involves constantly working relationships between scientists and engineers of all sorts. NASA and ESA are primarily, ESA being the European Space Agency, are primarily focused on exploration with Mars at the current time. And there is a series of missions that are designed to search for evidence of life on other planets and in planetary material delivered to Earth. That's really our two ways of trying to find out whether life exists beyond Earth. We either go out and look for it, or we look for it in rock material that has been delivered to Earth. My third bullet point, astrobiology is a leading international peer-reviewed journal thanks to Marianne Liebert Publishing, and we've been fortunate enough to be able to continue ramping up this journal over the 12 years of its existence as we go into the 13th year. So what are we looking for when we're looking for life on Mars? Really, if we take a look at the Earth's fossil record, we know that there's fossil evidence for life fairly early in Earth history. The Earth is about 4.5 billion years old. We see evidence in rocks 3.8 billion years old for life, microbial life. So if we think about what Earth was like at that period of time in Earth history, between about 4.1 and 3.8 billion years ago, it was a period of time called the Late Heavy Bombardment Period. This is the part in our planet's history where it was large enough that it was pulling in and sweeping up all of the material in its planetary orbit. It was accreting and growing in size as it swept up material in its orbit. And every time these impactors hit, basically they would create a lot of havoc on the Earth. They did things like delivered water and gases in terms of asteroid impactors. They also brought a lot of heat energy to the surface of the planet. And that resulted in constant changes to the surface of the Earth in terms of where life, if it did emerge at that time, where it could actually exist and thrive. So you have a rather hostile period of time, about 4.1 to 3.8 billion years ago, when we actually know that life did emerge under probably the most worst conditions it could have, yet it survived. All planets accrete by planetary accretion in a similar way, cleaning up material in their orbit. Early Earth had a history that was very similar to early Mars. So the question is, if life emerged on Mars like it did, can we find evidence of it? And if it didn't emerge on Mars, why not? You could say this about any planet, but we focus on Mars in particular. It's our closest planet, and it had a history very early, very much like ours. So we have missions going to Mars, and I actually we have the Mars Science Lander mission that's currently on the surface of Mars. It's launched, as Marianne said, last November. We were fortunate enough to be able to be there. I would recommend, if you've never had an opportunity to see a launch in your life, go see one. Well, it was very moving. And when I started to think about all the men and women involved and what it took to put that rocket up there and to get a spacecraft to land on the surface of Mars, it's just, it blows my mind away every time I think about it. This is a high-resolution digital image that Curiosity has taken of itself in the last couple days. If you go to the NASA website, there is a lot of information about this mission, so I'm not going to go through a lot of that, except to say that there has been a new announcement by NASA just yesterday that in 2020 we're going to fly another mission to Mars. We're going to take leftover parts from Curiosity. We're going to have another competition for a new suite of instruments, and there will be yet another mission going to the surface of Mars that will go in 2020. So this is an exciting new opportunity for scientists involved in astrobiology. So what are we looking for when we're looking for evidence of life on another planet? Basically, we're looking for suites of what we call biosignatures. I categorize these in one way, three main categories of biosignatures. This is evidence for life either in the form of microfossils or extracellular remains. You're looking here at a piece of rock that's been fractured. You're seeing filaments that are on the order of 0.5 micron diameter in cross-section sticking out of the rock. You can also look for microbially influenced sedimentary structures. So you can look for things like stromatolites or fabrics in rocks. Anytime there's microbes living in a natural environment and they're attaching to the surface of the rock, they're contributing to the formation of that rock if in fact the rock is accreting at the same time. They may be directly involved in precipitating minerals. Sometimes they're just involved in influencing where the detrital grains lay and contribute to the structures such as these. These are things on the order of, stromatolites on the order of about 1/2 meter tall. There's other types of chemical fossils that you can look for, things like biominerals that are produced directly by bacteria, biomarkers, lipid remains, stable isotope fractionation, chirality of different types of molecules. The point is that there are many varieties of signatures that we could look for by life, that life forms, but we want to have multiple lines of evidence for those because they tend to corroborate each other. And even more importantly, it's critical to have some idea of the context in which you find these types of objects, because without context, geological context, it's very hard to decide whether something actually is evidence for life. In fact, what NASA is planning in the future, potentially as early as, the next couple of decades, is a sample return mission. We can send fairly sophisticated suites of instruments up to the surface of the planet to look for possible biosignatures. But we also know that really to get the highest resolution, to be able to apply multiple techniques that can't fit on an instrument platform or can't be miniaturized, we need to bring samples back from Mars. And that is, in fact, something that's on the docket. And one of, I think, the major motivations for sending another mission up there in 2020 is to get the types of samples that we want to bring back to do further laboratory studies on. There's a couple of different perspectives for why, for things that we need to keep in context when we're looking for life. One is the science perspective. We want to know if life did emerge on another planet. Is it out there? Or are we, in fact, the only example of it? Because we do have the problem of one, right? We're it, as far as we know. So when we're out there looking for evidence of life, we're looking for evidence of bacterial life, we're looking for multiple suites of biosignatures, and we're looking for those in some sort of geological context, in an environment in which life could have lived. Another perspective for planetary science exploration is the issue of planetary protection. And that has to do with the fact that as we send instruments from Earth out to other planets, we have a responsibility to make sure that we don't contaminate other planets in terms of taking something from Earth to another planet. We also have responsibility when we bring an instrument or planet back, or not planet, an instrument or a mission back to Earth, that we don't contaminate Earth. So all of these perspectives are being incorporated not only in the future missions that are going on with space agencies, but also in the idea of a Mars sample return mission and the type of facility that would be involved in actually housing return material and allowing us to analyze it. Also, there's two different types of approaches when you're looking for evidence of life. Are you looking for evidence of fossilized life or evidence of living organisms? Fossilized evidence, if we go back to our biosignatures page, basically you can find evidence of microfossils, you can find microbially influenced structures, and then you can find things like biomarkers and stable isotopes if there's carbonaceous material preserved. You have a much wider suite of evidence that you can look for and analyze and characterize if you're actually looking for living organisms, right? So you can imagine, because of this difference, that you would have two very distinct types of instrument platforms depending on what type of mission you're sending. So the fact is, it's not really possible to search for fossilized life and evidence of existing life all in the same instrument package. We're doing our best at this point in time, and they'll continue to improve that as new instruments come online and can be miniaturized and fly. But in fact, if we were to bring different types of samples back, we would have a better opportunity to make those types of distinctions and apply the proper instrument analyses for looking for different types of life. I should say something that Vicki had mentioned to me a while back, Vicki Cohn at Leapfrog Publishing. It is interesting. We are going to Mars and we're searching for life, yet we're not allowed to go into those regions of Mars where we would expect to find any evidence of living organisms, where you might actually have liquid water, because we don't want to contaminate those regions. Those are special regions, and so we need to stay away from those environments right now until we can ensure that we aren't going to contaminate Mars. So we're looking primarily for fossilized evidence of life. We need to have a geological context and actually find the evidence there in that geological context that we would expect based on what we know about what life can do and how it lives on Earth. There's been two major biology breakthroughs that have had a significant impact on astrobiology. You're all familiar with the 16S ribosome RNA tree. It's generated out of, this is one that's adapted after something that Norm Pace had put together. At about the same time, Carl Stetter and Thomas Brock were actually culturing thermophiles and hyperthermophiles from high temperature environments. I want to focus the rest of the talk really on life at high temperatures, how we search for that life, and how that plays back into an astrobiology search strategy. The reason I'm motivated to do this is when I look at this tree, this is a temperature scale. 90 degrees C is the highest, the red lineages. Here's our position of the hypothetical last common ancestor. When we looked at the lineages clustered around this root of the tree, or hypothetical last common ancestor position, what we see is that these are really organisms that do live at high temperatures. And so the question is why? Did life originate at high temperatures? Or is it just that life that lived at high temperatures survived during that heavy bombardment period in Earth history? So remember, 3.8 to 4.1 billion years ago, probably earlier, the Earth was getting pelted. If life emerged on the planet, and if it emerged in a water-rich environment, which we would expect was distributed on the ocean floor, every time that you had a huge impactor come in and vaporize the ocean or vaporize part of that ocean, you would put a lot of stress on the system, right? Basically, the argument goes that it's very possible that the only life that could survive during that period of time was those organisms that lived at the highest temperatures in and around hydrothermal vents on the ocean floor. And so maybe the reason that we see a tree like this is not because life originated here, but because it was the only type of life that survived during that period. We still don't have the answer to that, but in any case, what we do know is that these ancient high-temperature organisms were in fact very, very early inhabitants of our planet. I have no idea of what time it is. Can you help me with that?
2:57.
2:57, okay. Let's just focus on a couple of main ideas here. If you're developing a search strategy to go to another planet, you're looking for evidence of life in places that you're familiar with on Earth. This is a hot spring from Yellowstone National Park. Here's your boardwalk for scale. And what you see here, this is Grand Prismatic, the largest hot spring in North America. If you haven't been to Yellowstone, I encourage you to go. This is the microbial mats that form in the outflow channels of a hot spring. As you move away from the highest temperature near boiling parts of this hot spring along one of these outflow channels, you're essentially walking an evolutionary timeline in Earth history. You're starting off in the center of this pool at near boiling temperatures with the chemolithotrophs, those organisms that live they get their energy and carbon sources from the solution, and they're basically living at life's upper temperature limit. You move out into the next, you see kind of a bullseye pattern here, the distribution of life. You next move out into non-oxygenic phototrophs, ultimately into lower temperature oxygenic phototrophs, and then ultimately into marshes. So it really is like walking an evolutionary timeline. The reason this is important is because hot spring environments not only contain a large diversity of microbial life, they concentrate a large diversity of microbial life in a very small area. They concentrate it in an environment where really you have a primary mineral forming. In this case, it's silica. Silica is well known for preserving fossilized evidence of life. And in fact, we can find silica-rich deposits on Mars, and they have been found on Mars. So, and there aren't too many places where you can actually form high concentrations of silica in very concentrated areas. So you've got a real high chance when you find a hydrothermal deposit that's monomineralic, like silica, that formed under hydrothermal conditions, to determine whether life ever emerged on the planet. We would expect that those lineages that formed at high temperature, if life emergence on Mars mimicked what it did on Earth, to be something that would be possible to find. Whether you would find higher order taxa as you move away from that environment really isn't known, but obviously you're at least looking at different windows of life history throughout a planet. And so it's a great opportunity for looking at that. I would just say mineralizing systems like hot springs or hydrothermal environments, they have the right ingredients for preserving evidence of life as well. Several environments do that. The idea is that if you have microbes living in this environment at a hot spring, that's great because you can look at the diversity, physiology, et cetera, ecology, learn more about those systems. But if they're living in an environment where minerals are precipitating out of the water, whether the life is there or not, then you have an opportunity to preserve evidence of life. So this is two examples. Basically, you're looking down at a petri dish size object of a microbial mat that's forming streamers. Here's the lithified equivalent. Life can be preserved, evidence of life, in fact, morphological evidence that we can see visually, can be preserved in these types of environments. If morphological evidence, visual evidence is preserved, ultimately there's multiple signatures that can be preserved in these environments. I won't go through the details here. I think we want to stay on schedule. I've got probably a half a dozen more.
5 minutes?
5 minutes? Okay. The hot springs are a terrestrial analog in which we're looking for life, and I've tried to give you some of the reasons why, because of the diversity of thermophiles, the high preservation potential because it's a mineralizing system, it's detectable from space because it's monomineralic. If you look at some of the structures around, these are SEM images, I'm not going to go through them all, but just to say that there are structures that are very distinctive that form around hot spring environments. Depending on which community you're in, remember if you walked along that thermal gradient, you're going to walk through different communities of organisms. Depending on where you're at, you can see various structures that line up with what kinds of activities those communities are involved in and how they leave information in the geological, either through cellular remains that become preserved because the organisms are attaching to the surface, or because this is actually one of these structures that's in cross-section. What you're looking here at is this type of a surface, which is on one of these structures, in cross-section. Here's where you see the filaments actually sticking out. But you've got an interplay of processes by which the biology is there on the surface of the rock as it's growing, as it's accreting. And then you also have periods of time where you have no biology present, which essentially caps that information onto the underlying layers. And you start this dance again with biology coming in, attaching to surfaces, becoming preserved. This interplay actually is very favorable to preservation. And so there's a lot of opportunity for preserving organic remains and doing a lot of detailed analyses of the types of objects that can be preserved in these environments. If we look in the geological record on Earth at hydrothermal deposits, ancient hot spring deposits, this is a rock about 360 million years old or so, we actually find evidence when we look at the morphology of this hand specimen that indicates that it formed at the higher temperature ends of the system. At any one time in the high temperature end of the system, around the edges of a hot spring, you may see a deposit that looks like this. These are microspicules that are on the order of 10 millimeters or less diameter and with a very small relief, on the order of 5 millimeters or so. If you break a couple of them off, you see that they look like this. They have a very interesting layered structure, very conical shape. In fact, when you look in the geological record, what you see are structures like this. They look very long, but really what these are successive layers of structures like this that have continued to grow for a long period of time. Okay? If you were to look at any one growth layer, you would see structures like this. But it continued to accrete. And structures like these are preserved. So the ability to go to an environment where you have a geological context, where you can recognize based on the mineralogy that it was hydrothermal, the ability to go and try to put together the paleoecology, look for evidence of the structures that are produced or influenced by the organisms, different types of organisms, gives us an opportunity to use hydrothermal environments as astrobiology search sites. I just want to finish here by saying that we have found silica-rich hydrothermal deposits on Mars. We found it both in our rover mission. Our Mars exploration rover mission found silica-rich deposits. And in fact, we found it with our orbital data missions, very distinct silica-rich deposits that are in fact associated with volcanic activity. This is important. We need to know where to go if we're going to search for life. It's not just a matter of going and searching in special regions where you have water. If we're looking for fossilized life, we're trying to look for very specific environments that we know could have supported microbial life. And the ability to find now evidence of silica-rich deposits that are probably associated with some sort of hydrothermal activity associated with volcanic setting gives us great reason to think that we have a shot at going there and looking and assessing the astrobiology potential of these types of deposits and look for the types of features that we are familiar with when we look at these extreme ecosystems in their modern settings on Earth. Space exploration missions are important not only because they bring together people in multidisciplinary ways. They're also an international effort. There are joint missions between NASA, ESA, other types of other space agencies around the world. Our current focus is on Mars. That's not necessarily a bad thing. We've put a lot of effort and money into Mars. It is our closest planet, and it would be really great to see some successful accomplishments with these missions as they go forward. And we're seeing and hearing about them now almost every day or every other day. New things that are being done on the surface of that planet that we haven't done before. So it's very exciting, and I do encourage you to check out the NASA website for new discoveries that is happening because of curiosity. Astrobiology does address one of these fundamental issues. You know, are we alone in the universe? If not, what's out there and what do we need to do to make sure that we don't interfere with it? If we are the only ones here, in fact, maybe that's a little bit more compelling reason for why we should take care of our own planet. It's a great way of getting science into the minds of kids. Most kids are intrigued by space exploration. NASA's done a great job of reaching out to them. NASA's also reached out a lot to the public worldwide through its website and interactive opportunities there, and then also a lot of teacher professional development. So I think in one aspect of NASA is looking at how they are campaigning the efforts that they're doing to get science into the hands of the public. I think one of the most exciting things is that hopefully we're going to continue as a country to have space explorations for many, many decades to come. We're on the road to making great strides on Mars. We will continue to expand exploration into our solar system. Ultimately, we are shooting for human exploration of our solar system and potentially beyond, depending on what the technological advances are. The idea that we could actually find evidence of life elsewhere is a question that's been around for a long time and one that obviously is not going to go away, and it's been great to be involved in helping move that field forward. Thanks.